Okay, so now let's look at the subtopic 10.1, measurement of growth. Okay, so first of all, for this subtopic, firstly you have to know the definition. Okay, so macam yang saya dah explain tadi, what is growth? Growth means irreversible permanent increase in quantitative parameter such as size, volume, length, height or surface area that can be measured. So remember, for growth, okay, it will involve, okay, irreversible permanent increase in quantitative parameter. So remember, quantitative means the parameter that can be measured. Okay, and then what is development? So development is defined as a qualitative concept that involve differentiation through which unspecialized cell or tissue change in form, shape and degree of complexity for specific function. Okay, so kalau dari segi development, okay, kita lihat dari segi qualitative concept. So apa qualitative concept? Dia akan melibatkan differentiation. Differentiation means bila berlaku differentiation of cell or tissue, okay, so dia akan menjadi specialized, okay, based on their specific function. Okay. Now, we look at how growth is measured. So, for growth in unicellular organism, how growth in cellular organism is measured. Okay, so due to their small size, so growth is measured by counting the numbers of individual present in culture at specific time interval. So since untuk unicellular organism, the size adalah lebih kecil, so macam mana kita nak consider growth that involve the unicellular organism? By counting the number. Maksudnya kita tengok dari segi bilangan individu yang ada di dalam setiap ataupun di dalam sesuatu culture at specific time interval. Okay, and then unicellular organism reproduce asexually through binary fission for population growth to take place. Means that how unicellular organism is reproduced, macam mana dia nak tambahkan bilangan, okay, adalah melalui asexual reproduction. Contohnya melalui binary fission. Okay, binary fission means belahan dua-dua. Alright, then we look at the growth in multicellular organism. For multicellular organism, okay, the growth of multicellular organism have three phases. The first one are cell division, second cell enlargement, and then third are cell differentiation. Okay, so now we look one by one. Okay, the growth in multicellular organism. So firstly, what is cell division? Okay, cell division, it will increase in the number of cell through mitosis. So means that macam mana bilangan sel tu boleh bertambah adalah melalui proses mitosis. Okay, next we look at cell enlargement. So what is the meaning of cell enlargement? It will increase in cell size and volume, and then increase in cytoplasmic content and number of organelle. Another one, each cell grow to its own limit. Okay, so bila kita tengok dari segi cell enlargement, dia akan melibatkan pertambahan dari segi cell size dan juga volume, dan juga increase in cytoplasmic content and also the number of the organelle. Okay, so for cell enlargement, it will different in both plant cell and also animal cell. So for plant cell, it will involve uptake of water by osmosis into large central vacuole, and then fresh material is supplied by photosynthesis. For animal cell, it will involve assimilation, which is the cell get new material from digestion. The keywords here is digestion, and synthesize new molecule. Okay, and also it will synthesize protein-rich cytoplasm. Alright, so another one, cell differentiation. Okay, so what is cell differentiation? Cell will undergo change in form. Why? To perform specialized function. Another one, to get the same set of genetic information to produce a diversity of cell type. Okay, so for the example, apa yang kita katakan sebagai cell differentiation? Contohnya, if vascular tissue undergo differentiation, so it will be differentiated into, one is xylem. Okay, so the function of xylem is to transportation of water and also minerals. Another one, vascular tissue also can differentiate to become phloem, which is its function is transportation of product of photosynthesis and hormone. Maksudnya the keywords here adalah apa yang akan berlaku kepada sel bila saja berlakunya cell differentiation. So the cell will specialize to perform the specific function. Okay. And another one, cell differentiation in human. So adult cell divide repeatedly, resulting in a group of cell, and then cells start to differentiate from each other in its structure and chemical composition. Okay, so now we continue with measurement of growth for multicellular organism. Okay, so the growth involves increasing of protoplasm of an organism. So actually, what is protoplasm? Protoplasm is the living part of a cell that is surrounded by plasma membrane, which includes cytoplasm, nucleus and other organelle. So means that kalau berlakunya, kita kata increasing of protoplasm, so kita consider dah berlaku growth in the multicellular organism. Okay, then growth is measured by recording the changes in parameter over a specific time. So the parameter that commonly used are, okay, for example, the parameter that used to measure the growth are biomass, width, height, number, length, size, volume and also the surface area. Okay. Next, parameter for measurement of growth in multicellular organism. So how the parameter is measured? Okay, either we measure by using the wet mass, okay, or dry mass. So what is the difference between wet mass and also dry mass? So wet mass is the mass of organism in normal condition without internal water remove. So means that kalau untuk wet mass, kita ambil kira dari segi mass of organism, okay, without internal water remove. Maksudnya without remove of water content inside of the organism. So what are the advantage by using the wet mass to measure the growth of the organism? It is easy. And then second, no need to kill the organism. And then third, growth can be measured repeatedly. Okay, but the disadvantage, okay, wet mass is not accurate. Why it is not accurate? Because the water content in organism is fluctuated. Maksudnya, dari segi mass of organism, kalau kita gunakan wet mass, okay, dia akan dipengaruhi oleh water content inside of the organism. But what is the difference with dry mass? So dry mass is the mass of organism after its water content is fully eliminated. Maksudnya kalau untuk dry mass, okay, apa yang berlaku pada organism adalah kita keluarkan water yang ada di dalam organism tersebut. Okay, so implies actual growth of the organism. Okay, so what is the advantage for dry mass? Okay, for dry mass, the measurement is more accurate. But for disadvantage, organism are killed. Why kill? Sebab kita nak removekan the water content inside of the organism. So organism tu akan dibunuh dulu. So growth cannot be measured repeatedly. So means that the same specimen cannot be repeated sebab organism tersebut dah dibunuh. Okay, and then second, need large number of specimen. Okay, and then third, time consuming. So dia akan ambil masa yang lebih lama compare dengan wet mass.